Thank Professor Liu for introduction. Good afternoon, ladies and gentlemen, and it is my honor to be here and talk about epidemiology and evolution of carbapenem-resistant Klebsiella pneumonia. Why I focus on the Klebsiella pneumonia, Professor Liu had mentioned Klebsiella pneumonia has a very thick capsule. It also causes nosocomial infection disease, including pneumonia and bloodstream infection. So in my presentation, I would like to share with you our recent group work on the epidemiology findings. I also introduce some evolution of CRKP, and also talk a little bit on the strategy. As we know, in this year, in May, WHO updated bacteria priority pathogen list. You can see in the critical group, there are 2 carbapenem-resistant bacteria, including carbapenem-resistant Echinopelmani. It also includes carbapenem-resistant Enterobacterales. And we know Klebsiella pneumonia belong to Enterobacterales. And also, 2 months ago, WHO also gave a warning on the antimicrobial resistant hypervirulence Klebsiella pneumonia. It mentions for the hypervirulence Klebsiella pneumonia can cause severe infection in healthy individuals. It also causes the disease is insidious, and also mentions the emergence of AT23 isolate with resistance to last line antibiotic like carbapenems were reported in several countries. So WHO gave recommendations on the laboratory diagnosis capacity for the early and reliable identification, including the molecular methods for detection for virulence gene and resistant gene. So let's look at what is HVKP. Hypervirulence Klebsiella pneumonia were first reported in 1980s. It's originally in Taiwan and marked by the cryptical liver abscess. And you can see the colony is very mucoid. It shows the hypermucous viscous. And also it can cause very severe infections, including multi-site manifestation and metastatic. It also includes pneumonia. Here it shows the endophalitis. Usually HVKP is susceptible, but resistant strain, including ESBL producing isolate and carbapenem-resistant isolate, will emerge and even cause outbreak in hospital. So there are 3 patterns of formation or transmission of CRHVKP. The most important pathway is go this way. You can see the Klebsiella pneumonia can acquire resistant plasmid, including the plasmid carriage KBC and NDM, and become resistant to carbapenem. Also, it can acquire virulence plasmid and become to CRHVKP. Another pathway is a typical HV clone, including AT23. It's usually susceptible, but it can acquire a resistant plasmid and become to CRHVKP. Also recently, a study had reported a single plasmid with virulence and resistant gene, but this is very few reports. So in our hospital, we organized the CRE network in China in 2011. Now, for the recent year, our CRE network spans over 90 hospitals across 28 provinces in China. Overall, we have 10,000 CRE strains. So I will show some data on our CRE network. So first of all, we can see from 2011 to 2021, among almost 6,000 CRE strain Klebsiella pneumonia accounted for the highest proportion of at 70%, followed by E. coli at 16%. And in this group, you can you can see the most common infections is the respiratory tract infection. It accounted for 46%, followed by bloodstream infection 18%, and urinary tract infection 16%. Overall, 2,000 CRE strains were sourced from the ICU. and with more than 1,600 of these being Klebsiella pneumonia. So our colleague also investigated the association between CRE strain and the patient group and the regional difference. You can see here this orange red is stand for KPC. You can see the KPC is the most predominant resistant mechanism, followed by NDM, this green one. And also, we can see the different thing is for the children. You can see here in the CRE among the patients under 18 years old, the NDM is the primary resistance mechanism. We also investigated the MLT for all of the CRE strain. Our analysis have found 4,000 isolated reviewed the presence more than 200 ST. And ST11 was the most prevalent, followed by ST15. And ST23 only counted less than 1.3%. We also did phylogeneous and phylogeographic analysis at T11 CRKP. You can see here in the global, IT emerged in 1996 on the American continent and spread globally and diverged into sublineage with local populations. And also, you can see here at T11 expanded in China during the mid-2000s, and initially dominated by the KO47. And by the 2010, were replaced by KO64, spreading from southeast coast to the inland. And also, we found a KO, for this figure, you can see here, the green is stand for KO47, and the blue one is stand for KO64. Very surprisingly, you can see KO64 significantly increased from 3% to 45%. And the right figure in here, you can see at T11 KO47 is a green, dark red. It predominant in 2015, but recent years at KO64 predominated this blue one. So our study has found that at T11 KO64 had become the new dominant subclone. Also very surprising, you can find that at T11 KO64 carries more virulent gene. Even carries 3 virulent gene and even 4 virulent gene. Altogether, 30% of the KO64 carry resistant virulent gene is much higher than KO47. Also, we do the global phylogenetic tree of AT11 CRKP. You can see here for B and C, it belongs to KO47. And for clade E here, is more resistant and carries more resistant gene. Our colleague also performs the clinical data review, and our data has shown that the mortality of the patient greater than 64 years old patient was over 50%. It's very high for this group, much higher than other clades. We also perform some in vitro study, and for here is figure B and C and D. You can see that the clade E has a stronger resistance to phagocytosis, and also competitive growth advantages and potential survival benefits, and direct competition and antibiotic stress. Also for GVAR study, we have found clade E with specific SMP. We named BOMs. They have 5 gene with SMP. We demonstrate that they have a notable propensity for expansion and phagocytic advantages. Another finding is about the AT11's virulence plasmid. We found KO64 when he acquired the virulence gene, it deleted 3 kb of the gene. That's why we don't know the reason. So our colleague performed the study. We do the gene knockout, and we complement it again. And we found when they delete the 3K, it can acquire increase antioxidant capacity and also increase survival in macrophage. So another pathway is about AT23, how it can acquire resistant plasmid. When it acquires a resistant plasmid, what kind of the adaptation it requires? So we also investigated. We found in our CRE network only 20 KPC producing AT23 carries the resistance. So it's very few, much lower than the AT11. However, when AT23 acquire a KPC resistant gene, very surprisingly, its capsule gene, WCG, were inserted by IC KPN26. When the gene WCG was lost, had the insertion, it also lost some virulence. Our mouse study has indicated that overall its toxicity decreased, and however, the survival rate increased. So we also in tracking the KPC evolution, we established a global database on the 15,000 isolate KPC Barry's isolate, and we over 20 years. Our study have dated the range of KPC transmission can be back to 1991, and revealed frequently gene exchange across countries. And the intra-Klebsiella pneumonia and cross-genial KPC transmission pattern varied across 4 distinct periods. Here, this slide shows the 4 stage. In the early stage, the few plasmid type were identified, and there no cross-genus transmission observed. However, in the second stage, a gradually increase in plasmid type. And in 2013 to 2017, a peak in dissemination. And maybe due to the COVID-19, in 2022, you can see the light reduction in plasmid members and diversities. And in the early stage, the intra-species transmission was mediated by the natural host syringe plasmid. However, for the late stage, and the cross-genial transmission was dominated by the wide host syringe plasmid. We also investigated the dissemination of MCR9. This is a colistin resistant gene within One Health framework. Our study has found that MCR9 initially spread from Western Europe to American and East Asia, and then the strain, South Asia and the Middle East gradually acquired acquired this gene. Also, the earliest MCR gene likely originated in human and subsequently spread to animals and the environment. We also have the question in our collection. We found MCR one very located in the E. coli. However, MCR9 is a preferred Enterobacter colloqui, so we don't know why. So we do some searching and found maybe a codon usage bias is a very important reasons. In this work, we found MCR 9 carriage plasmids prefer the nucleotide TC over T compared to MCR1 carriage plasmid among the Cinnamonas, the codons. So when we do some amino acid replacement by the codon change from C to T, we can find the increase gene expression in E. coli significantly higher than the Enterobacter cloacae. So maybe they can explain the reasons of the relationship between the gene and the bacteria. Also, we all know diagnosis is very important for infectious disease control, especially in AMR pathogens. So based on the metagenomic from the third generation sequencing, we developed a rapid track database to track the resistant bacteria. We construct a clustering pan-genomic database. We also incorporated the sequencing alignment and similarity of the taxonomy algorithm. We also developed a visual website. This website is user-friendly. You can put the NGS data into this website. It can show you the string typing and the evolutionary work, and also have some other prediction AMR data. Also, we cooperated with some researchers, including Beijing Normal University and Hunan University. We want to develop some new drugs, such as peptide and palliative cage and photosensitizers. We hope to do some invention work for the AMR strains. So my conclusion today is maybe we have some strategies, including the combating AMR based on One Health approach. We also need ominous work to investigate the sources of resistant gene and the transmission mechanism. We also need to develop rapid diagnostic techniques for pathogens, and including the new invention works. That's all. I thank the CRE network. We also thank the NSFC and other MOST for grant supporting. Thank you very much.
Questions?
Thank you very much for your talk. I would like to know if you noted a change of the mutants producing less capsule with respect to the infectious site. I was thinking to Ernst that described in Nature Microbiology that encapsulated can be more sticky, strained, and be more urinary tract infection prone, and instead the other decapsulated are more sepsis causing infection, systemic infection more often. Have you noted if this change toward the less virulent means also different kind of infections?
Yeah, it's a very good question, and we try to, in my clinical practice, to develop some method to rapid diagnosis or detection the subclone. Because they have the difference, the phenotype and the molecular background. So it's a way to do that. We hope we can find some POCT method. We'll try this especially for our findings for BMPS, also for hypervirulence gene, to find some way to that. Thank you.
Okay.
Thank you very much. It's a very impressive collection of isolates that you have and being able to see this. I just want to ask you about the clonal replacements. Do you have any hypothesis why that happened? Or is there any event that is associated with that clonal replacement, or any changes in patient population, etc.?
Yes, we are thinking about that. Why the clone replacement? Sometimes I think maybe it has some balance between the resistance and the virulence. Maybe it's a little bit like the COVID. It becomes less virulent in the population. But for the AMR, for Klebsiella pneumonia, I just thinking about maybe antibiotic pressure is a very important factors, and also for other factors, because in our collections we all know the baseline is very important. The AT11 compared to AT23. In my presentation, in my opinion, I think AT11 carries the resistant gene, the KBC and other DM. Maybe he has some advantages for the survival of advantages at the antibiotic the pressures. So for this way, if he want to evolve, maybe he also can in this population he can also acquire some virulence gene. So it's complex actually. Yeah.
Any more questions? Okay, let's thank Professor
Thank you.
Wang again.